All right, engineers, in this video we're going to talk about the pancreas, but specifically we're going to focus on the activity of glucagon. Okay, so if you remember, if you guys watched our video on insulin, go back if you guys haven't, watch that video on insulin, get caught up on what was happening with the pancreas, because we're just going to go ahead and dive right in. But just as a quick review, remember the pancreas is a heterocrine gland, right? And so that means that it's actually made up of two components. We told you, right, that one was the exocrine, which was the acini. That was the one that was accounting for about 99% of the pancreas. It was the one that was making the pancreatic juice, and which was rich in digestive enzymes and bicarbonate. The other part was the islets of Langerhans. Okay? So these are the two parts of the pancreas. These are the two components. And if you remember, we divided the islets of Langerhans into two cells, the alpha cells and the beta cells, right? Those were the two components. Now, if you remember, we said that the beta cells, that was the discussion for the insulin, they were the ones that were responsible for secreting insulin. And if you remember, insulin was stimulated by hyperglycemia. Hyperglycemia, right? So it's anything greater than about 120, maybe even tops 130 milligrams per DL. Whereas the alpha cells, they're the ones that are secreting glucagon. And glucagon, its primarily stimulus is actually going to be hypoglycemia. And again, you can remember that by that little thing I told you, that it's released whenever glucose is gone, whenever it's low. And generally anything below 70, maybe even a little bit below that, milligrams per DL. So if you remember, what was that range? The normal range is about, we like to give it about 80 to 120. That's like your general range, okay? For every average individual. Now, like I said, anything below 80 and maybe even a little bit below 70, right, is a primary stimulus, and it's hypoglycemia, right? So that would stimulate these alpha cells to secrete glucagon. And again, how do you remember alpha cells make glucagon? Because there's an A in glucagon and an A in alpha, okay? There's no A in insulin. Then, if you remember, anything greater than 120, maybe even a little bit even 130, is hyperglycemia, and that's a stimulus for insulin. We're going to talk about primarily what then? What's the stimulus of glucagon? It's when glucose is gone. So it's hypoglycemia, right? There's low glucose levels. So if there's low blood glucose levels, hypoglycemia, what does that mean? That means that very little glucose is going to be coming into this actual alpha cell. Okay, so it's transporting this glucose into the alpha cell. Now, the certain literatures will say that this actual transporter, it's not GLUT4, it's technically GLUT1, okay? Whether or not that's significant, it's not really important in this conversation. I'm just giving you a comparison as to where it was insulin, right? Insulin specifically in the pancreatic beta cells had those GLUT2 receptors. So let's say that it's GLUT1 specifically on the alpha cells, but neither here nor there, not too important. That glucose, though, if you remember, gets converted into pyruvate, right? And then if you remember, pyruvate gets taken into the mitochondria and converted into acetyl-CoA that undergoes a lot of that Krebs cycle activity. Then it gets taken to the electron transport chain, those NADHs and FADH2s, and produces ATP. But if there's very little glucose getting into the cell, you're not going to really make a lot of ATP. But here's where it's a little funky. You see these channels here on the membrane? These channels right there? They're sensitive to that ATP. So whenever there's actually ATP, when there's very little amounts of ATP, these channels are really sensitive to charge. So they're not really going to respond to a lot of like charged particles. So whenever it's when there's a lot of glucose coming in, inside of the cell, there's a lot of potassium ions that accumulated, if you remember. In this case, it's a little different. When ATP binds here, where there's very little ATP, these channels are very, very sensitive. They still actually close. They still close. And potassium ions are still going to be blocked from exiting. But it's not going to be so many potassium ions accumulating in this area that it's so large that it causes a significant membrane depolarization. There's going to be less membrane depolarization. If there's less membrane depolarization, this calcium channel is going to be more sensitive to that. And it's going to open up and allow for calcium ions to start moving into this vicinity. So let me explain this one more time. Very little glucose makes very little pyruvate, which makes very little acetyl-CoA, which makes very little ATP. Now, this decreased amount of ATP due to decreased glucose levels, these potassium channels are very sensitive to ATP, but you don't want to produce too much ATP, because if it's too much ACP, it's going to affect these channels. Another thing is that these channels are really, really sensitive to charge. If the inside of the membrane becomes too positive, these membrane channels become really, really weird. Okay? So you don't want there to be too much of a membrane depolarization. So when ATP binds, it still closes those potassium channels and potassium accumulates, but it doesn't cause as much of a significant membrane depolarization. And then these calcium channels, due to that not a significant amount of a membrane depolarization, opens and calcium flows in. Why am I saying this? Because guess what? If your glucose levels are really, really high, you make too much pyruvate, you make too much acetyl-CoA, you make too much ATP. Too much ATP is going to bind onto this and keep it closed very, very strong and tightly. So then a lot of potassium is going to stay into this cell. And what's going to happen to the cell? It's going to become extremely positive. Remember what I told you. When it's really, really positive inside of the cell, guess what it does to that calcium channel? It closes it. And calcium can't come in. I'm going to explain why that's important. But again, remember, we're talking about low glucose levels, low ATP, less sensitive effect on the potassium channels. Potassium ions do accumulate, but it's less of a significant effect of membrane depolarization, which causes these voltage-sensitive calcium channels to open, and the calcium flows in. In the meantime, remember these actual pancreatic alpha cells are synthesizing this glucagon. So it's going to be like pre-synthesized. Now there is other stimuli that can affect glucagon synthesis and secretion besides this actual low glucose. One of them is, they believe, norepinephrine or epinephrine can actually, so your sympathetic nervous system, so SNS, sympathetic nervous system, they believe that this can be a stimulator of specifically glucagon secretion, okay? That's one mechanism. They also say that it could be sensitive to cholecystokinin, which is a hormone produced by your intestine, and secretin, which is another hormone produced by your intestine. They say that they're also stimuli. Neither here nor there, though. Just to let you guys know, there are other stimulators of glucagon production rather than just low glucose levels. But this is the primary stimulus. Okay? So now, if you remember, let's say that this is a specific gene, and that gene is getting transcribed to make mRNA. That mRNA then goes, and it actually goes to these ribosomes that are on the rough endoplasmic reticulum, right? So it goes out to this area. And then if you remember, what is it going to do? It's going to synthesize this peptide, right? And this peptide is going to get pushed into the rough endoplasmic reticulum. If it gets pushed into the rough endoplasmic reticulum, it's actually forming proglucagon. It'll get cleaved and modified, and then it'll come out in this little vesicle from the rough ER. And then you're going to have in this little vesicle, you're going to have that glucagon hormone, right? Then that glucagon is actually going to go to the Golgi. And in the Golgi, it undergoes a little bit more modification reactions, and then what happens is it undergoes specific types of packaging. And then look what happens to it. Out of the Golgi, look what comes out of the Golgi. That nice little vesicle, and that vesicle contains glucagon. So you could have multiple vesicles out here. I could have, look, I'm going to draw 3 vesicles just for the sake of it. So now what do I have out here? I have vesicles containing the glucagon hormone. What did I tell you? If there's less potassium that's actually staying inside the cell, there's less of a significant membrane depolarization, and it's just the right amount of membrane depolarization, in other words, becoming more positive in the cell, that causes these calcium channels to be very sensitive and open. When the calcium comes in, what happens? Remember, there's proteins here on this membrane, and there's proteins here on that vesicle. What calcium does is he links these together. He helps them to migrate and fuse with that. When this vesicle fuses with that cell membrane, look what happens. It fuses and opens up into the extracellular environment. If it opens up into the extracellular environment, what is it going to do? It's going to release out what? Glucagon. And glucagon gets pushed into the bloodstream. And we just synthesize glucagon, okay? Now, again, why am I telling you that this whole thing with the ATP? Because if your blood glucose levels are really, really high, you're going to make too much pyruvate. Too much pyruvate makes too much ATP. Too much ATP causes these potassium channels to become even really, really sensitive and accumulates too much potassium inside of the cell. When there's too much potassium accumulating inside of the cell, it causes the cell to become extremely positively charged. And when there's an extreme positive charge, that inhibits this calcium channel. Calcium can't come in. If calcium can't come in, can you actually allow for these vesicles to migrate and release glucagon? No. So that's why low glucose is a stimulator, but high glucose is an inhibitor of glucagon secretion. And again, remember that CCK stands for cholecystokinin, secretin, which are gastrointestinal hormones, norepinephrine and epinephrine are also stimuli of glucagon secretion. Okay, so now we made our glucagon. And again, let's recap. What was the glucose levels in the blood? They were really, really low. Now, primarily, when are your glucose levels low? Okay, besides having like a pancreatic insulinoma, you're generally going to have low glucose levels in a condition which is called the fasting state. Okay, so your fasting state meaning that your post-absorptive state. So in other words, you haven't eaten in a while, okay? So you haven't eaten in a while. This is generally the time whenever your glucose levels are low, physiologically, not assuming any type of pathological conditions. If your glucose levels are low in the blood, that's not good because your brain depends on glucose and so do other tissues. So we want to be able to get the glucose in the blood up. How do we do that? Let's see what glucagon does. First thing glucagon does is it comes over here to the liver. It loves to act on the liver. So let's say here's this glucagon represented by that black dot. Here's our glucagon, okay? And he's ready to help. What does he do? He binds onto this G protein coupled receptor. And you guys should know this mechanism by frickin' memory now. It stimulates a G stimulatory protein. G stimulatory protein is normally bound to GDP, which keeps it off, but then when it's bound to GTP, it's turned on. Goes and activates a effector enzyme. This effector enzyme is actually called adenylate cyclase. And then adenylate cyclase takes ATP and converts it into cyclic AMP. And then cyclic AMP activates protein kinase A. Okay, why is all of this important? Because you remember, glucagon is a peptide hormone. He can't pass through the cell to exert its effects. It's got to make second messengers. So, how does he do this? Now, if you remember during the actual fed state, we had a specific molecule that we actually synthesized. If you guys remember, it was called glycogen. So glycogen is just a polymer of glucose, right? So this is our glycogen molecule. So let's call this again. This is called glycogen. The first thing that this glucagon is going to do is it's actually going to help to convert this glycogen specifically into the basic building blocks of glycogen. You know what the basic building blocks of glycogen is? It's called glucose. What is it doing? How is it working? You know there's specific enzymes. We'll talk about this in biochemistry. There's a specific enzyme called glycogen phosphorylase. But protein kinase A is stimulating that enzyme and causing it to break down glycogen into glucose. Why is that significant? Because where can that glucose then go? Look where it goes. Into the blood. If it goes into the blood, what happens to the glucose level? So now these glucose, if you're bringing a lot of glucose into the bloodstream from that glycogenolysis, which we're going to name, what's going to happen to the glucose levels? It's going to increase. So again, what is this reaction here that just occurred? This reaction here is called glycogenolysis. Gnarly. That's pretty cool, right? Now, what else can happen? Glucagon's also pretty cool because not only does he promote glycogenolysis, he uses other molecules. So before we keep going, let's show what he does on the adipose tissue because it's going to be related, okay? So glucagon comes out here, and what he does is he acts on this receptor. This is adipose tissue, so this is your fat tissue, right? So again, who is this guy right here? This is your glucagon. He's excited and ready to help. What is this glucagon? Let's actually make him a little bit happier. He looks like a little angry. All right, so glucagon, what is he going to do? He's going to activate this receptor, this G protein coupled receptor. So again, G stimulatory, binds to GTP, which activates him and turns him on. When that happens, he goes and activates an effector enzyme. If you remember, that effector enzyme is actually called adenylate cyclase. When it stimulates and activates adenylate cyclase takes ATP and converts it into cyclic AMP. And then cyclic AMP activates protein kinase A. Again, why is this important? Because glucagon can't just get into the cell and start exerting its functions. It has to do it through a second messenger. Now, why is that important? You guys have probably seen this 1,000 times already. Look what I got here. You see that enzyme right there? This enzyme, look at him. He's ready to roll, okay? This enzyme is ready to roll. This enzyme is called hormone sensitive lipase. Guess what protein kinase A does? He comes over here and he'll phosphorylate that enzyme, activating the enzyme. And you know what that hormone sensitive lipase is going to do? It's going to start cutting specifically those ester bonds that hold the triglycerides together. And then when it breaks that up, it's going to break it into its two constituents. What are the two constituents of this triglyceride? So this triacylglycerol, or triglyceride, TAG, right? It's just another word for saying triglyceride, or triacylglycerol. It's going to break it into its two components. One is glycerol. The other is these fatty acids. Okay, so the other is fatty acids. So these are the two components that are going to be broken into. So this glycerol is really, really critical. Let's bring this glycerol over here now, and it's going to come into this liver cell. Now, what happens with this glycerol is that protein kinase A, he's going to work through special types of mechanism. Look what he does. He comes over here, okay, and he acts on the specific enzymes. And there's specific enzymes that help to be able to convert that glycerol into glucose. Specific enzymes. So you can take the glycerol, turn it into DHAP, and then shift it upwards to make glucose. You know what else it can do? You know there's other different types of molecules that are present inside of the liver that are really, really important. A lot of other different types of molecules. This one, we can say are called amino acids. Okay, so you can have amino acids in the area, or you can have glycerol in this area. Look what that protein kinase A also does. He also says, okay, I'm not only going to stimulate that pathway, but I'm going to come over here and I'm going to stimulate this conversion of amino acids into glucose. And it also stimulates this. It undergoes transamination, and then after that it gets turned into maybe a specific keto acid or into pyruvate, and then it gets converted into glucose. So again, let's look and see what this guy's doing in the liver. One, he's activating protein kinase A. What is protein kinase A doing? Activating glycogenolysis, breaking down the glycogen into glucose by stimulating glycogen phosphorylase, right? It's also taking the glycerol, which was coming from the adipose tissue, and then stimulating the pathways to convert him into glucose. And it was also stimulating certain amino acids to undergo transamination and they get converted into glucose. Also, there is an exception here. If there's fatty acids that are odd chain, so odd chain fatty acids, just for those of you students who maybe want to know a little extra, these odd chain fatty acids can also get converted into glucose, okay? What is that called whenever you're taking glycerol, amino acids, and even lactic acid, and even odd chain fatty acids and turning them into glucose? That is called gluconeogenesis. Okay? Gluconeogenesis. So two really important effects here. One was glycogenolysis. One was gluconeogenesis. And what is this called when you're breaking down triglycerides into glycerol and fatty acids via the hormone-sensitive lipase? This is called, specifically, this event here, it's called lipolysis. Okay? So now, one last thing here. Glucagon also recently has been found to have an effect on the heart, specifically the myocardium of the heart. So let's say here's our glucagon. And here's, glucagon, and again, he's excited, he's ready to be able to help. He wants to be able to do whatever he can to get those glucose levels up. He also has another effect that is independent of the glucose levels. He actually does something really cool. I just thought I'd mention it. He can activate that G-stimulatory protein again, right? Which again goes and activates adenylate cyclase, which then actually does what? Takes ATP and converts it into cyclic AMP, and then cyclic AMP activates protein kinase A. What's that significant for? Protein kinase A comes over here and activates and opens up specific channels, these specialized channels. These specialized channels are for calcium, and calcium starts flowing in to this muscle cell. Why is that important? Because if you get a lot of calcium into that muscle cell, what's it going to be useful for? It's useful for that cross-bridge formation, for the contractions, right, to bind on troponin, change the shape of the tropomyosin, allow for the actual myosin to bind into actin, and then cause the sliding of those myofilaments. If there's more calcium than usual, won't there be enhanced contractility? Yes. So what's the effect that glucagon can have on the heart? He can increase contractility. What is that called when a certain agent can increase contractility? It's called a positive inotropic agent. Okay? So if he increases your contractility, he can technically increase your stroke volume, your cardiac output, maybe even your BP. But to come back to this, if you actually have glycogenolysis, right, that's going to get glucose into the blood. If you have glycerol, amino acids, lactic acids, odd chain fatty acids getting converted into glucose via gluconeogenesis, and that glucose that you form gets into the blood, what happens to the glucose levels? You're going to bring those glucose levels up because you're accumulating a lot of glucose. And you're going to bring those glucose levels up to where you can get it right within that range, again, of about 80 to 120 milligrams per DL. That would be the desire. Because then once you start getting it too high, what will happen if it gets too high? You guys remember? If it starts to get too high, what happens? If you get a lot of glucose, it makes too much pyruvate. A lot of pyruvate will make too much ATP. Too much ATP will affect these potassium-sensitive channels to the ATP. If too much ATP, it'll cause a significant membrane depolarization. And if it's too membrane depolarized, it inhibits the calcium channels. And then no calcium comes in. If calcium doesn't come in, can you cause the release of glucagon? No. And then glucagon doesn't get released. Who does get released if it's too high? Insulin. And it's a beautiful way of having this effect called antagonism. Antagonism is when two hormones are opposing each other. So glucagon wants to be able to do what to your glucose levels? Bring it up. Insulin wants to do what to your glucose levels? Bring it down. There's also synergism. If you could think about synergism, it would be other hormones that are performing the same function. So for example, glucagon has almost the exact same functions as epinephrine. So those two are synergistic to one another. They love to be able to increase the blood glucose levels. And then there's one last term called permissiveness, which is when one hormone needs to be present in order for another hormone to function. Okay? And that could be a perfect example of thyroid hormone. Thyroid hormone is really necessary in order for certain types of steroid sex hormones to cause brain development. Okay? But I just thought I'd throw that at you again. All right, Ninja Nerds, in this video we talked about a lot of information. I really hope you enjoyed it. I hope it made sense. If you did like it, hit that like button, put a comment down in the comment section, subscribe. We really want to hear from you guys. All right, Ninja Nerds, until next time.